Today's Saturday talk, we are having Mr. Merwyn Dcosta. He's a M.Pharm graduate from the Bombay College of Pharmacy, Santa Cruz, which he completed in August 23 with good grades. And he did his B Pharm in St. John's Institute, Palghar in 2020. He has got three research internships. First one he did in ACG as an associated where he was in the quality control department. He did his second internship in CI Cipla, where he did an assessment of pharmaceutical products and optimizing the activities and costs there. The third institute where he was in the DHIO, Research and Engineering. He did his internship, demonstration of techniques for preparation of silver nanoparticle induced films and in silico technologies employed in the computational calculation and modeling of graphene sheets. He has done a lot of research projects in various areas, including machine learning, artificial intelligence, and development of novel molecules and treatment methods against breast cancer, as well as in silico screening of phytochemical naringin. Okay, and almost done 9 research projects in various areas, pharmaceutical sector. He has published few publications, and he has got excellent academic credentials in form of DBT scholarship from the government of Maharashtra when he did his bachelor's. And I'm glad that we have a good qualified person in the form of Merwyn Dcosta in our today's Saturday session. And I request Merwyn Dcosta to take the forum and proceed with the talk.
Good evening, everyone. My name is Merwyn Dcosta, and I'll be talking a little bit on artificial intelligence and machine learning in drug discovery today. And let's start with today's topic. So, as the topic suggests, the name suggests, artificial intelligence, machine learning in drug discovery. As there are already qualified people and knowledgeable people in the meeting, we know everything about artificial intelligence and machine learning. Let's see something about drug discovery. So, as the name suggests, let's see first what is drug, and then we'll see what is discovery. So, in the pharmaceutical terms, we quote the term drug to any substance which is intended for diagnosis, treatment, cure, or prevention of a disease. Anything. For instance, we take crocin when we have fever, so paracetamol becomes the drug. We take some Ayurvedic formulations like Triphala for constipation, so that becomes a drug. So anything which we use for diagnosis, treatment, or prevention is called a drug. Then, as we already know the word drug, let's see what is discovery. So, as the name suggests, discovery is a process where we find a new drug on a new target or an existing target. We very well know both of these words. But do we understand why do we need drug discovery? Why do we need to discover a new drug? Where there are almost thousands and lakhs of drugs which are marketed these days in the market. Why do we need a new drug? So let's take a small example. For instance, we use paracetamol when we have pain or when we have fever, but still there is ibuprofen in the market. So sometimes it is quite weird where we have two molecules doing the same thing. Even ibuprofen does the same thing, paracetamol does the same thing. So then why do we need ibuprofen when we have paracetamol? So the main twist in this drug discovery, or the word drug discovery, takes a podium when we take into consideration what the body does to the drug. For instance, when we have paracetamol, there are different things which the body does to the drug, and when there is ibuprofen, there are different things what the body does to ibuprofen. So that is the reason, simplifying this statement, we can say paracetamol treats pain in a different way, ibuprofen treats pain in a different way. So each molecule has their own pros and has their own cons. So focusing on drug discovery part, the process of drug discovery takes around 10 to 15 years. So then still, when the process is so long, when we have when the procedure for drug discovery is set over the period of years, then why do we need artificial intelligence and machine learning in this drug discovery procedures? When we have everything which is set, when we know we have different molecules for different type of approaches, what is the need for AI and ML to come into picture? So we'll see the next slide to understand why we need AI ML in drug discovery. So if you can see this slide tells us about the conventional drug discovery process and the process for drug repurposing. Now, what is drug repurposing? Drug repurposing is, for instance, instance, if we have heard about, we all have heard about aspirin. We all know that aspirin is a very good painkiller and it has been used for years. But few of us know that aspirin is also used to prevent heart attack. So the activity of aspirin to prevent heart attack was done using the approach called as drug repurposing, wherein we try to find out what is the other activity of the drug other than its main activity. Now that is okay. Drug repurposing is okay, but not all drugs have other allied good activities. So then again shift our focus to conventional drug discovery. Now a normal drug discovery procedure takes around 10 to 15 years. It takes 10 to 15 years of time for this drug discovery procedure, whereas companies invest a lot of money, a lot of time, labor, scientists invest a lot of efforts, and after 10 to 15 years, the assurity of a single molecule becoming a drug is just less than 20%. So when AI and ML took their hold over the international market, or the international, we can say international scientific literature, scientists thought, why can't we use this advanced machine learning technologies or advanced machines computers to expedite this procedure and to increase the chances of a molecule becoming a drug. That is the reason the concept of artificial intelligence and machine learning in drug discovery procedure came to existence. And this was, I guess, artificial intelligence has machine learning has been into this drug discovery procedure from the late 90s, I guess. Now let's see something about drug discovery procedure and what is drug discovery procedure exactly. So when we say ki, okay, now for instance we need to discover a new drug for example breast cancer. So what do scientists actually do when they start discovering a drug for breast cancer? First is target identification. Now what is target identification? Target identification is a process when scientists first decide which disease they want to work. So we select breast cancer. So we start working on breast cancer. Now after we select the disease, we scientists try to find, or rather I can say we try to find what are the enzymes, proteins, or I can say what are the body functionalities which cause breast cancer. So after they find out which are the body functionalities, then they see which proteins on a smaller level cause breast cancer. After they know a protein, you can actually say that we have identified a pinpointing biological molecules linked to a disease. After you find the protein, okay, for instance, there is this X protein which causes breast cancer. Then you see that are there any molecules which are produced by the body which either inhibit that protein or they accelerate the activity of the protein causing breast cancer. After all this study is done, you can say your target identification process is complete. After target identification, the second important step of the drug discovery process is the hit discovery. Now, what does this hit discovery mean? Hit discovery means when you develop a target. For instance, you say, okay, X protein causes breast cancer. Hyperactivity of X protein is causing breast cancer. So you need a molecule which will depress or which will suppress the activity of X protein. So in hit discovery, what was conventionally being done was scientists used actual pen and paper, and they sat down and they prepared, or I can say they developed lakhs of analogs of a molecule. For instance, we say ki, okay, a benzene ring with a 5-member carbon is giving a good inhibitory activity against the protein. So scientists sat down and they designed lakhs of molecules. They screened this lakhs of molecules for different potential properties. For instance, the solubility of the molecule, is the molecule permeating the biological membrane, and all this was done experimentally. Now you imagine lakhs of molecules being screened over such vast parameters. It took a lot of time. It took a lot of time, lot of efforts, lot of money pumped in, a lot of scientific minds occupied in just one disease, I can say, and the assurity of what the molecule from these lakhs of compounds becoming a drug was just less than 20%. So then after still scientists did this the conventional way, and they screened the molecule, and lakhs of molecules were screened, and then some thousand molecules which passed the parameters was taken to the next step called as lead optimization. Now in lead optimization, what they did was they checked the efficacy and safety. Now what is efficacy? Efficacy is, in simple terms, efficacy is how well your drug is showing activity. For instance, we say paracetamol, we take crocin, which is 650 mg. In 650 mg, within one night your fever goes down. So that is the efficacy of paracetamol. So in lead optimization, scientists check the efficacy for all the 1,000 compounds which has come down from hit discovery. Then they even check the safety of the compounds. Safety as in they'll check whether this molecule is actually safe. Is it showing any harmful effects on your liver, kidney, or for instance, you are targeting breast cancer, and is that are these compounds, thousands of compounds, showing any other hazardous activity? So they check it using some lab test, or they can check it using some animal models. These were checked. These were done on a very extensive and exhaustive scale on lead optimization. After doing some these lead optimization studies, the next step was preclinical trials. Now in preclinical trials, what was actually done was there was a filter done in the lead optimization step. So the thousands of compounds were brought down to hundreds of compounds. Now these hundreds of compounds which were taken into the preclinical trials were then tested on animal models. In preclinical trials, we usually use mouse, rats, some ape-like animals like monkeys, to check whether the compounds are actually safe before testing it on humans. But the brutal part of this preclinical trial is the compounds which are used cause a lot of fatality in the animals, and lot of animals are dead during this preclinical trials, and a lot of animals face a lot of side effects due to those untested compounds. Because of this, there is a lot of death of the animals, as well as there is a lot of failure which is faced in preclinical trials. Still, after doing all the preclinical steps, there are some two-figured molecules, like some less than 100 molecules, which are then further taken to clinical trials. Now, in clinical trials, these compounds are actually tested on human volunteers. They are tested on human volunteers to see if there are any side effects, or what is the dose at which the compounds needs to be given to the humans, such that will act as a medicine and that will treat breast cancer. But the problem with all these 4-5 steps, initial drug discovery steps, is it is time consuming. It is time consuming. It is capital consuming, as well as it requires a lot of scientific expertise. And the problem is engaging a lot of scientific expertise and in just one uncertain process was very problematic to the pharma companies. So what they did was they thought, why don't we use computers in just a second. Why don't we use computers in these 3 steps? We club these 3 steps using computers, AI ML methodologies, and we do the hit discovery, lead optimization, preclinical trials in computers, and we take down the compounds which showed promising results in these to the clinical trials. And the surprise to this was the time period from 10 to 15 years was dropped down to 5 to 6 years after employing AI ML methodologies actually in drug discovery process. And the fatalities caused to animals was also drastically decreased, and the results in clinical trials were very promising after employing artificial intelligence and machine learning. So pharma companies as well as academic institutions started getting their interests more in the computational in silico aspect of the drug discovery process. Now, to quote an example here, we all have heard about ranitidine. Ranitidine is was actually a very blockbuster antacid medicine which was marketed by a pharma company. But recently in 21-22 mids, it was withdrawn back. It was taken back by the company because of implications from the FDA, because ranitidine was found to have a very lethal impurity, and the metabolite of ranitidine was known to cause a lot of problems with the liver. It was known to cause a lot of problems with the liver, and the company realized it very late. After years of marketing of ranitidine, they realized it in 2022, 21, 22 that ranitidine was actually toxic for people who were taking the drug. So at this point, if there would have been artificial intelligence methods which would have been implicated in the initial steps, I feel ki this would not have happened, and we could have realized the part very much earlier before even taking the drug to the market. This slide shows some basic steps involved in steps involved in basic research. Now, what is basic research? In this basic research, as we saw in the previous step, basic research involves everything from designing a molecule to the preclinical phase. Now, if we now what conventionally used to be happening is scientists used to design lakhs of molecules. Then it used to go for hit discovery. Then the molecules were filtered. Then it used to go for lead discovery. Then again the molecule used to be filtered. So these procedures used to take a lot of time. So what when AI/ML started getting integrated in life sciences or drug discovery procedure, what happened was scientists took a molecule. They for instance identified the target. Okay, so for example, we want to work on breast cancer. They identified, okay, this molecule is well known for its anti-breast cancer activity. Now, when we go for finalizing or designing the analogs, now in a chemical molecule, there can be infinite number of analogs. But what scientists do is, previous to actually designing the analogs, they take this molecule, they take the protein which is associated with breast cancer, and they dock these two. After docking, what happens is the AI algorithm tells you, okay, this is the molecule, and in this molecule only R1, R2 and R3 is responsible for the activity, which means if any one of these R1, R2, R3 is missing, the compound will not show any anti-cancer activity. So scientists get a predetermined I can say a pre-known knowledge about the product, that when we start to actually build analogs, we try replacing only R1, R2 and R3. The central structure can remain intact because this does not contribute to any anti-cancer activity. And then we can again screen it in silico, and only the compounds which show potential or promising activity against cancer will be then synthesized and subjected to preclinical and clinical trials. Now, preclinical and clinical trials cannot be ruled out totally after employing AI/ML models, but we can reduce the number of casualties, or we can say the amount of time invested in clinical and preclinical trials. So that can be done using after integrating artificial intelligence, machine learning in drug discovery procedures. Now, this slide, we will see something about how AI will actually play a role in drug discovery procedures, how AI can help us in drug discovery. So the first step is target identification and validation. So as I told you earlier, this is if you can see the picture here, this is the picture of a target, how a protein in your body looks like, how a protein in our body looks like. So what scientists do is, scientists tell the AI algorithm, okay, this is my drug of interest. I want to see which target this drug binds. So the AI algorithm goes, it checks for any checks for all the databases which are present, and it tells us, okay, these are the targets which the molecule binds. So with the help of AI, scientists can do these procedures in minutes. Earlier, the steps which were taking years can now be done in minutes. For example, if I quote an example at this point, if we take into consideration Alzheimer's disease. Now, Alzheimer's disease is mostly caused due to the formation of amyloid plaques in your neurons. Basically, the neurons get blocked. So what was happening is, earlier when scientists started doing their research on Alzheimer's, it took them 2 to 3 years to find out why Alzheimer's did happen. So in 2 to 3 years, they found out, okay, Alzheimer's was happening due to formation of amyloid plaques. But it took them 20 years to understand why that amyloid plaques were forming. So AI can play a very crucial role in this part, minimizing the time frame. The second utility of AI is virtual screening of potential drug candidates. So what artificial intelligence can do is, after scientists make lakhs of molecules, they design lakhs of molecules in computers, they can tell the artificial intelligence algorithm that, okay, screen the molecules for XYZ parameters. For instance, in pharmaceuticals, we take the permeability of the molecule under consideration, we take the solubility under consideration, we take its degradation potential in stomach enzymes. So what that AI molecule will do, AI algorithm will go, it will take all the lakhs of molecules as an input, it will check the structural parameters, it will check the electronic properties, it will check all the possible parameters, and it will give you some thousands of molecules or some hundreds of molecules which will pass the parameters, and we can then use those hundreds of molecules for further preclinical and clinical trials. So the steps like hit discovery and lead discovery are totally replaceable by AI algorithms. The third important role of AI algorithm is predictive modeling and simulation. In predictive modeling and simulation, what we can do is, we can take up a protein, we can take up a molecule, and we can simulate it in a computer. We can create the conditions which are in your body, the salt concentration, the osmotic pressure, the ion concentration, and we can do a dynamic simulation which will help us to understand what is actually happening to the molecule after it is going into the body, without actually administering the molecule into your body. So this is a very beautiful applicability of computers, I can say, implicating AI and ML algorithms. The fourth role is in streamlining experimental workflows. Now, after scientists do all the initial steps, at a point we have to synthesize the compounds. At a point we have to synthesize the molecules. Synthesizing the molecules also comes with a lot of problems and troubles, because synthesizing a molecule takes a lot of time and a lot of experimental reagents and a lot of optimization procedures. So AI models, there are existing AI models which tells you the synthetic accessibility of a compound. Synthetic accessibility means how easily the molecule can be synthesized. There are AI models which tell you. There are already AI models there is a web server called as Swiss Expacy. It is developed by Swiss Institute of Bioinformatics. They actually have these AI models which will tell you how well or how easily the molecule can be synthesized. Secondly, there are molecules which will actually give you a synthetic pathway, which will give you an experimental procedure to how to actually synthesize the molecule. So this is also a very beautiful applicability of AI. And the last is personalized medicine. Now in personalized, to explain you in very simple terms what is personalized medicine is, an example of breast cancer. What happens is, in breast cancer, there are two types of breast cancer. One is a normal one and another one is triple negative breast cancer. Now the problem is there are a lot of treatment for normal breast cancer, but those normal breast cancers, though the normal molecules against normal breast cancers don't work on triple negative breast cancer. So AI models can help us to analyze. There are a lot of databases wherein it is already reported, okay, these are the biomarkers present in triple negative breast cancer. So we can tell AI to actually go and find out the biomarkers and what could be a possible and a potential treatment for TNBC, instead of actually sitting and experimenting on the TNBC triple negative breast cancer patients. So this can be done using AI. This is a very important slide wherein you can see milestones of applications of AI in drug discovery. In 2012, AlphaFold was introduced. AlphaFold is the subsidiary company of Alphabet, Google parent company. So this is a very robust and a very versatile tool to actually predict protein structures. Then in 2016, Atomwise launched the AI model. 2018, IBM was into AI. 2019, Exentia developed its first AI designed drug, which is now in clinical trials. And in 2020, the most important part of all the human history, I can say COVID-19, where we wanted an expedited drug for COVID-19. And still, there are applications which are going on in the field. These are some of the branches where AI has been well established, like analyzing data analysis, molecular structure visualization, clinical trials, and drug repurposing, which we have already seen. The last part is applications of AI in pharmacovigilance. What is pharmacovigilance? Pharmacovigilance is post-marketing surveillance. 50% of the medicines which are in the market are under post-marketing surveillance. Post-marketing surveillance means the company still takes data about how the medicine is acting in us when we actually are being treated on a disease. After the marketing, after FDA approval, still 50% of the drugs in market are in post-marketing surveillance. So AI can help us do a faster analysis of the data, automate the procedure, improve signal detection, and basically do everything what a human could do in a post-marketing surveillance. So AI can totally replace humans in post-marketing surveillance. Now, after we have seen all the advantages of AI, let's talk something about disadvantages. Why is AI ML still not that prominent in drug discovery? First is data quality issues. Data quality issues means the quality of data generated in health, life sciences is still not that great or not that understandable by the machine learning models. Second is integrity complexities. Integrating AI into well-established workflows, well-established drug discovery workflow is still very difficult, and it requires a lot of staff training. Third is ethical considerations. Not all pharma companies have a very open approach to AI, because most of them think that AI is more of a data piracy and potential biased kind of a thing, because the machine would not think as good as a human thinks. And the fourth is evolving regulatory landscape. Still, the regulatory authorities have not devised guidelines on how the AI should be implicated, or at what extent the AI should be implicated. Hence, there are certain barriers to applications of AI ML in drug discovery. These are some of the applications other than drug discovery wherein we can use AI in pharmaceutical product development, clinical trials, QA, QC, manufacturing. We'll skip this and go to the case study. These are some of the organizations which have been using AI right from the 1900s, late 1900s. Coming to the case studies, these are the case studies which are well defined in the literature. I'll tell you some case studies which I have worked on. So these are the three case studies which I have worked on. Firstly, I have used AI ML models to develop AI. I have used AI ML to develop algorithms which will actually predict the odor threshold of fragrant molecules. What is odor threshold? Odor threshold is whenever you have any fragrance around you, or you use a perfume, there has to be a certain concentration of the molecule in air, after which you will get the odor of the perfume or any odor active molecule. So I have developed models which can tell you whether the molecule has an odor or at what concentration will it have odor, using artificial intelligence and machine learning methodologies. And I have got good results. I have achieved an accuracy, a correlation coefficient up to 0.8%. 80% of my model is predicting the odor threshold well. Secondly, I have used it for developing lead compounds against tuberculosis. And third is used it to investigate phytochemicals against Alzheimer's. These are some of the case studies which I have used AI on. You can see these graphs. These graphs are from my own research. This dark line is the actual values. Actual values are the values which are actually predicted by scientists, which are actually taken up by scientists by doing experimentation. They have performed the experimentation and got those values. And the yellow line shows the values predicted by my models. This is from one of the model, and this is from another model. So we have got good results, and I have fine-tuned the models, and it is predicting the odor thresholds well. And with this, I guess we conclude with the presentation. And I thank you everyone, JanyuTech, for giving me the opportunity. And I conclude with this. Thank you.
I have two
Huh.
questions actually. First,
Yes.
so right now, like for any either diseases or vaccinations,
Huh.
those particular medicines, like not all humans are able to cope up with those medicines. So
Right.
sometimes it reacts with their body,
Right.
right? So
Right.
using AI and ML, is it possible for us to customize a particular medicine for a particular disease for a particular person? Yeah, that's what, like for any diseases, just an overall overview. I just
Huh.
wanted to know about it. Is it possible?
Sir, actually, it is been done. As if you could see, I gave you an example of precision medicine, and the
Yes.
very versatile topic of breast cancer. Now, in triple negative breast cancer, if you see, the drugs which are available on breast cancer now in market don't work on triple negative breast cancer. So, AI models, or I can say AI algorithms, are actually being employed, and precision medicine is in use, whereas scientists are developing precision medicines for TNBCs, because in TNBC there is a very tricky thing called as breast cancer stem cells. So what this breast cancer stem cell does is, it after you take out, for instance, in breast cancer, we remove the breast if the cancer is widespread. So in
Yes.
if the patient has triple negative breast cancer, what happens is this triple negative breast cancer cells get converted into breast cancer. So AI models are being employed to understand these triple negative breast cancer cells, to see what are the biomarkers on these breast cancer stem cells, and to develop models and to develop molecules which are actually very specific for those stem cells. So it, yes, it has been used.
Okay. And my second question, it is something like which I feel like it's a future scope for us. But what I wanted to know is like in few diseases, just like tuberculosis, that like the second stage, MDR, multiple drug resistant tuberculosis.
Huh.
I have been told that this particular disease, they have an impact on your DNA. So
Yes.
it is somewhat like if a particular person is having that particular disease, and if their descendants might have the chances to get it.
Okay. Uh,
So my question would be using AI. Yeah, yes. So my question is, using AI, is it possible to, remove those diseases from the DNA, like for the betterment of the future generation?
Haha.
Is there any research going in that area?
Actually, sir, multiple drug MDR tuberculosis is more related to the bacterial DNA rather than the host DNA. Malab, for if you see, there are drugs which actually target the bacterial DNA, but the thing which you told, that it affects the host DNA. I worked on tuberculosis sometime back in my masters. There are some research which are published, but that is not widely accepted by the scientific community, because there are a lot of speculations in MDR. MDR is more related to the bacterial aspect, because what is happening is, in MDR, the bacteria takes up the anti-tubercular drug, but then it actually using some channel based pores on its surface, it throws it out. So it is not exactly related to the DNA part of the host. But if you say at the DNA part of the host, there are AI methodologies which are implied, or I can say applicable in genomics. So people are using scientists are using AI in genomics. They are using AI to develop CRISPR-Cas technologies, and they are using AI to know what gene editing should be done so as this disease can be taken off from the body. Yes, it is done.
Mr. Merwin, I have a question.
Yes, sir.
As you told that the medical council or the regulatory authorities are yet to accept or we can say bring into regulations the use of
Yes.
AI
Yes.
into the various stages of the drug development. Is it applicable like even at the preclinical stages or at the clinical stages? Which stage they have not actually, they are not brought or accepted AI?
Sir, it is more towards the clinical trial stage and the FDA regulation stage. It is widely accepted at the preclinical stage, because we use techniques like molecular docking. Molecular docking mimics the preclinical stages. So at preclinical level, lead optimization, hit optimization, AI is used.
So as long as any practical humans are not affected, medical council allows you the freedom to use AI. right?
Yes. Yes. Yes.
Okay. Is it is it the same thing with the international regulatory authorities also, or only with the Indian?
No, sir. Yeah, international. Because more of the integration of AI into drug discovery is happening in the US and Europe rather than in India.
Which means that the Indian companies have not fully started
Yes.
using AI. That's what it
Yes.
means.
And not exactly. Most of the time, most of the research in drug is being done in the academics part rather than in industries in India. So academic institutions are using AI, sir, but still it is in development phase. Like there is a lot of problems with this AI. You have to give it a lot of data, and people in India are not that open with sharing their data. That is the most problem, because even I have faced the same problem. While developing the AI models, you need a lot of experimental data to train your models, and in India we do not have the culture of sharing our data, even in academics mostly.